Hello everyone. Good news. This is the last video series for the year, so very exciting times. And I'm so proud of you guys for making it this far and going through all these fabulous videos. You are amazing. All right, so now we're on chapter 17, and this is dealing with transcription and translation. There are a lot of details that you're going to need to remember as you go through this. So let's take a look. This should be review. We did go over this last year, but the details we don't really touch on in general bio. So here's the basic understanding. This is what we call the central dogma of biology, that things go from DNA to RNA to a protein. Now let's go back to an analogy I use often. The DNA that is in your nucleus is like a large cookbook, but it's the original copy of the cookbook that we have to protect. And inside that cookbook are thousands of recipes. Those thousands of recipes represent the proteins that make your body work. Your DNA, when you make a copy of it, you need to have the entire recipe book. However, if you go in and you want the recipe for chocolate brownies, you don't also need the recipe for spaghetti and meatballs, for country fried steak, for fried chicken. You don't need for every single recipe in that book. You don't need all those. You just need the recipe, the one page out of thousands and thousands that codes for the chocolate brownies. That's what happens when we talk about base protein synthesis, okay? And when we discuss transcription and translation, what is happening is that we're going into the cookbook, which is your DNA, and we're pulling out one recipe. And we're changing that recipe, just like you would with a cookbook, from a paper copy to, in this case, DNA would be the paper copy, to mRNA, and that process is called transcription. And you can see it right here. Here's your DNA, and you go from DNA to mRNA, transcription. And then you go from mRNA to protein, translation. And let's look at the words. When you transcribe something, you copy it. And that's what you're doing when you are transcribing DNA and you're changing it from DNA to mRNA. They're both in the nucleic acid family. And now we're going to do translation. So we're taking mRNA and we're going to change it to a protein, something in a totally different macromolecule family. So that's why we call it translation. All right, so let's get into the details. Now I'm going to go ahead and skip this page. We're just going to really focus on the main points here. But you are welcome to read over it. What we're going to look at is how does a single faulty gene, this is an example, result in this albino deer coming on. So we're going to look at mutations today too, where there's errors in this transcription translation process. All right, so here we go. Before cell division, DNA has to duplicate. That's called DNA replication. We've gone over that in the last section, the last note packet. Now, sometimes after we've done, often after we've made a copy of the DNA and the DNA is sitting in the cell, we need to get the information out of the DNA. And that's when we're going to go into this transcription translation. Okay? And here's our central dogma. We go from DNA to RNA. That's called transcription. From RNA to a protein. That's called translation. Okay? And then what does a protein result in? A trait. It's a trait. So we take the DNA, we get a copy for eye pigment. We make that copy in mRNA, because mRNA is the copy. Then we take mRNA and we translate it through translation using tRNA and amino acids, and we create a protein, and that protein results in a trait. So let's say the trait was eye color. We get the eye color gene from the DNA, we make it into mRNA, and then we take that mRNA and we translate it into a protein that codes for the pigment in the color of your eyes. All right. And what we're doing here is just going through these quick overviews. Transcription is the synthesis of RNA. Transcription uses mRNA. Translation is the synthesis of the polypeptide chain, the amino acid chain, under the direction of mRNA. And the ribosome is where translation happens. Now, the worksheet that you're given with the chart, I've had some people say, well, where is this found in the book? You need to go through it. This is found from you understanding the processes and being able to fill out this chart. You have to understand what enzymes are used for what takes place, where is its location. All of these details are important, and it's really a good idea to learn them in a chart form because then you can compare it. Okay, who are the major players in transcription? Who are the major players in translation? Versus who are the major players, or you're in biology, so it would be enzymes in DNA replication. All right, let's look. I'm going to go ahead and pause it, and then I'm going to pick this back up.